going to try to zoom through this as quick as I can. So hopefully we'll have a little bit of time for questions afterwards. If not, please jet me an email. I'd love to hear from you afterwards. So we've been hearing a lot about how climate change affects the way that plants grow. But in terms of the climate system, it's really important to also think about the way that plants break down, because it's the breakdown of plants that ultimately releases carbon dioxide back to the atmosphere, where it has an effect on global warming. So taking that really big scale perspective, most earth scientists think about plant decay in really simple chemical terms. You start with an organic carbon compound, you've got some enzymes, maybe they have a temperature dependent rate. Their activity winds up removing carbon from a system as carbon dioxide, where it hits the atmosphere and causes a lot of problems. But exactly how this happens really depends on where you're looking. So if we look in this particular beautiful habitat in coastal Manatee County, really closely at a suspended dam branch, closely enough that we use X-ray tomography to reconstruct the conditions inside a stem, you'll see a lot of fungal hyphae there. Those fungi are the organisms that are producing the enzymes, and it's their respiration that's responsible for a lot of the carbon loss from these systems. So at that really fine scale, as botanists, we're often interested in this interaction between plants, their senescent tissues, and communities of microorganisms that develop to break them down. But being in coastal Florida, it's important to recognize that's not the only process that's important. Let's take a step back, like a really big step back. And if we look from space, we can see that the same system can lose carbon through a completely different process. You can imagine that branch with all of its carbon and all of its fungi bailing off the tree, hitting the water, and then being flushed out to the Gulf of Mexico. So from that broad scale perspective, exchange between the terrestrial and the marine environment is an independent and really important way that this system is actually also losing carbon. So we have these two separate scales, very different processes, but they're not independent from one another. We can have broad scale temperature gradients that affect how these organisms work to break down carbon at the really fine scale. And interestingly, we can have fine scale processes like degradation of wood mechanical strength that increases the chances those branches are going to fall off and increases the rate of exchange. So a really simple equation turns into a very complicated picture when we start recognizing the fact that processes operate at different scales and those scales can interact. So what's a biologist to do? Fortunately, there is a ton of data about how scales interact. So conceptual foundations of hierarchy theory go back in ecology to the 1980s. The basic idea here is that you can have stuff that's happening at a fine spatial scale. Maybe those interactions are cool when you're looking at that scale, but they interact in a way that causes emergent features in the system. And you can have large scale processes that operate on those emergent features. All right, a lot of gobbledygook. Let's come up with a good schematic diagram of exactly what I mean. Okay, so consider an experiment. This experiment examines the relationship between wood decay rates on our y-axis and temperature in a study that looks at multiple sites across a big gradient. Okay, we can see two kinds of patterns that can pop up. The one that our simple chemical equation assumes is one that we can call scale invariance. What I mean by that is there's the same slope of the relationship between temperature and decay, whether you look within a particular site or if you look at across different sites within a region. Cool? So it doesn't matter really where you look. Spatial scale doesn't matter. But there's an independent process and one that shows up in a lot of decay studies, which we can call scale dependent. A scale dependent pattern in this case is one where if you look at the relationship between decay and temperature within a site, you can see a slope that's actually different from the relationship between decay and temperature across sites within a region. So in this sense, it's scale dependent, because if you're looking at the regional pattern, you can observe one slope. And that slope is significantly different from the slope that you would see within a particular region. And this kind of pattern implies that there are different processes that are operating. And understanding both of those processes and how they connect is necessary to get a holistic understanding of what's going on in the system. Okay, so that's our schematic diagram. What kinds of tools do we have at our disposal in order to analyze it? Hierarchy theory lends itself really easily to hierarchical models. Okay, so we can talk about different flavors of models. You have one model where every single one of your sites across your whole regional study is treated independently, right? You can also do what a lot of folks do, and if you're interested in those regional level patterns, you take all your different sites, you average them out, and you look at the relationship across all the different plots within your region, right? Neither one of those two approaches is entirely adequate for dealing with these kinds of hierarchical issues. Hierarchical models are way better. Hierarchical models let you take the parameters that describe the slope of the relationship at a local site as a function of parameters that govern the differences among sites within a region. So you can explicitly accommodate processes that are operating at those independent scales and actually measure how they're happening. So it's a really cool approach. All of this stuff, though, focuses on spatial scale. Oh, got lots of arrows. Let's zoom through those. Let's talk about temporal scale now. Okay, we're going to talk about temporal scale specifically in the context of wood decay. Now, why does wood decay matter? Well, there's a lot of wood out there. It's the second largest above ground carbon pool. And another reason why thinking about temporal scale is important for understanding wood decay is that it takes a long time. I've actually watched trees decompose. It's not the most exciting thing that you can do in the field, but it's really important. Right? The residence time of dead wood in most forest ecosystems spans from years to decades, right? And it's that process of that wood breaking down that releases all the carbon from these ecosystems back into the atmosphere. Okay, so understanding how that works really requires us to do cool experiments. But the experiments that we can do are just limited by practical considerations. They don't last very long. Right? You can expect that most of these are going to last as long as a typical grant. So we're talking 1 year, maybe 3 years. That's going to be the snapshot that you can use to understand what's driving patterns of wood decay. Another thing that's challenging is it just takes a lot of effort to get folks together in a lab to measure all the things that are going on, get all your covariates together. So most people will generally only study this at a single point in time. Those are practical considerations, but they have some really important consequences on the kinds of models that you can use to understand wood decay. Okay, so if we want to understand what drives wood decay rates, and we want to see whether or not models are adequate, what we're going to do in this situation is think a little bit about time. All right, so the most common model that people use is one that's kind of convenient for this style of analysis, one where you only sample once and you sample relatively early on. This kind of model assumes first order decay kinetics, right? Basically there's just one parameter. It's like radioactive decay. Right, so what's happening in the first time slice is representative of what's happening in a longer time slice. So it doesn't really matter when you sample your experiment. If you get it once, you get the information for that one parameter, and you're really good to go. However, we can sort of change one of these assumptions when we start thinking about the biological complexity that goes with wood decay. All right, so wood isn't like a radioactive isotope. Right? It's this complex biological tissue, and it's changing the entire time that microorganisms are interacting with it. Not only is the wood complicated and does it change, microorganisms themselves are really complicated. You can have different fungi that show up at the beginning, there's succession in that community, they change over to different fungi after all. Enzymes they're producing are going to be going crazy. Environments aren't super simple. We could have different kinds of soils, we can have different kinds of climates. You can imagine how all these things are going to interact. Right? So radioactive decay is probably not a good model for wood decay, and specifically not a good model because we can expect that the rate of decay changes through time with the wood in the communities that are breaking it down. So let's come up with one reasonable example, and we'll think about a functional trait. Maybe this functional trait is wood density. In this case, wood density doesn't change the overall rate of decay, but it changes how the decay rate varies through time. Right? So maybe it's going to speed up decay or it's going to slow down decay. In this particular case, let's assume that wood density accelerates decay. So there's like a lag phase. Start with really dense wood, decays really slowly at first, then you hit some kind of threshold, right? The wood's super porous, and then the decay rates really shoot up. Cool? So no constant decay rate. In this situation, you can get some super perverse things that happen in experiments. And they are perverse things that depend on temporal scale. Cool? So we've got a hypothetical experiment here with two different species. One that's got really dense wood, one that has wood that's not very dense at all. Otherwise, they have the same exact decay rate. If you sample this experiment a really long time in the future, you'll see what you would expect to see. The species that has relatively dense wood is going to be the species that has less tissue left in the environment, right? However, if you sample this experiment when about half the tissue is gone, you'll see no relationship between wood decay and wood density whatsoever. The perverse part is that if you sample this experiment right out the gate, you're going to get that opposite relationship between wood density and decay rates. You're going to get that opposite relationship because you're still in this lag phase. The wood hasn't lost enough of its density to really kick in. And you might imagine that this is going to give you like a super weird projection of how wood decay dynamics are going to play out in your ecosystem. And it's completely an artifact of the temporal scale of your experiment. Cool? Whew. running fast. So time scale really matters. All right, so we're gonna make a couple of predictions when we start thinking about time scale when we're studying wood decay. The first prediction that we're gonna make is that as this wood basically becomes soil, traits that really distinguish wood at first are going to become less important later on. And the second thing is, if we think that all this biology matters, and these models that are more complicated are important, we should be able to make more accurate predictions when we bring all of this information to the table. All right, so where are we gonna do this? We're gonna do this in my hometown, St. Louis, Missouri. There's some people here who love it, probably some people here who don't, but we worked just outside of St. Louis at Tyson Research Center. It's a oak hickory forest with a lot of really cool resources. In this forest, we had paired lot plots. So instead of your common garden experiment, we have a common decay experiment. We put wood from different species in the same spot, decaying under the same conditions, but those conditions kind of vary. And we look at a lot of different species. We have 21 different woody species at the site. They vary in a ton of different functional traits. And we're measuring all kinds of features of the environment, soils, microclimate. We're also measuring all kinds of features of our species, from wood density down to vessel length. Okay, we sample our experiment 1 year, 3 years, and 7 years into the future, and we're gonna analyze it using a cool Weibull model. I'm gonna zoom through this really quick. What the Weibull model is gonna give us is a new parameter that's gonna stretch or shrink decay through time. And we're gonna analyze it in a hierarchical Bayesian framework. What's cool about that is we can have each of these parameters vary with features of the environment or features of our individual species. Now we measured a ton of those features, and so we have to simplify our model somehow. These models are really flexible, so we can basically turn parameters on and off as part of the model. It's like super cool. I totally advocate this stuff. It just takes a little while to learn. And we fit all this stuff in R, which is a nice common framework where you can do a bunch of cool analyses. Awesome. 3 minutes? Sweet. Kind of. Thinking a little over. Okay, so let's look at our predictions, right? The first prediction was you start with really complicated and heterogeneous wood, you wind up with soil. So maybe you see a lot of traits that are important as first, but the longer you wait in decay, a lot of those things actually happen to vanish. So here we have all of our trait predictors, our environmental predictors are on the bottom, and we have different kinds of models and temporal sampling strategies in the columns. If we look at after only a single year of decay, there are lots of different wood traits that really influence how quickly wood decomposes. But if you look after 5 years of decay, there's only one, lignin, right? So all that rich texture that you saw, that really awesome discussion that you wrote about how vessel width is super important for decay, is only important as you look after 1 year, right? If you wait for 7 years, none of that stuff matters anymore. And also, the effect strength of lignin has fallen in half, right? So if you make a projection based on the strength of lignin in your study, you're gonna wind up with a super different concept of how that's going to influence residence times than if you wind up sampling your study a little bit differently. Environment, unsurprisingly, doesn't make that big of a difference. Wood traits are a lot more important. What's really interesting here is that there are 2 different traits that control actual time scale dependency in wood decay. Both wood density and vessel diameter stretch or shrink decay rates. And what this means is that if you only sample your experiment once, and if you don't wait long enough, you can get a really bizarre picture of how these traits actually influence wood decay in a system. So this is cool. Does it help us make predictions? We did an independent replicate experiment, and the punchline here is, yeah, it does matter. If you make projections based after only a single year of decay, you come up with a biased and poor prediction of how much mass is going to be lost from your species. But if you wait for 7 years, you sample multiple times, you improve your predictive accuracy a lot. But this is still an experiment. What happens if you go out into the field? We did that. At this place, there's a deadwood survey with lots of different species. And what we find is that just knowing what species it is and where it died, we can parameterize these different decay models and predict features of the deadwood. So for individuals and locations where we'd expect wood to have long residence time, they're more likely to be standing up, and they are in a less advanced decay stage, right? So we can actually use our experiment to predict features of the structure of naturally recruited deadwood over a 4-hectare forest dynamics plot. So that's all pretty cool. All right, so what are we going to conclude from this? The first thing that we're going to conclude is that because traits change decay rates, it's really important to set up one of these experiments so that you sample multiple times and you use one of these fancier models, right? Exponential is cool, but it's not going to let you predict things very accurately. Thanks, folks. And also, epiphytes are cool. We've got a new journal.